I chose to research the actin toxin. First, here is some background information. The actin cytoskeleton plays numerous vital roles in innate and adaptive immune responses and represents a common and attractive target for microbial toxins. It is not surprising that the actin cytoskeleton is one of the main targets of bacterial protein toxins, and thus of major importance for the host-pathogen interaction. Note that toxins are defined as bacterial products that can act in the absence of the bacteria, whereas the bacterial effectors depend on the presence of bacteria. An example of this is transportation into the target cells. In fact, there are at least 3 groups of toxin/effectors that directly modify actin molecules. The first group causes ADP ribosylation of actin at arginine 177, which in turn inhibits actin polymerization. The second group includes toxins that modify actin to promote actin polymerization and the formation of actin aggregates at threonine 148. The third group catalyzes a chemical cross-linking reaction of actin, thereby forming oligomers and blocking the polymerization of actin to functional filaments. The study titled "The Effect of Toxins on Inorganic Phosphate Release During Actin Polymerization" focuses on the second group, or toxins that modify actin to promote actin polymerization. First, what is actin? Our biochemistry textbook describes actin thoroughly, and so does this journal article titled "Actin as Target for Modification by Bacterial Protein Toxins." The article describes actin as a 42 kDa protein that is among the most abundant in cells. It typically accounts for as much as 10% of the protein in eukaryotic cells. Actin exists in 2 principal forms: globular, monomeric G-actin, and filamentous, polymeric F-actin. In other words, actin monomers, often called G-actin for globular, come together to form actin filaments, often called F-actin. F-actin is composed of 2 strands of linearly arranged actin subunits that are wound around each other, forming a helical structure. Each monomer is related to the preceding one by a translation of 27.5 angstroms and rotation of 166 degrees around the helical axes. It is important to note that each actin monomer is oriented in the same direction along the F-actin filament, and so the structure is polar, with discernibly different ends. G-actin is a flat molecule with dimensions of about 50 by 50 by 35 angstroms, as illustrated in Figure 1. Figure 1 gives the standard view on the flat face of actin. The structure of the actin monomer has been determined to atomic resolution by X-ray crystallography and has been used to interpret the structure of actin filaments, which are already somewhat understood through electron microscopy, studies at lower resolution. G-actin contains firmly bound 1 molecule of ATP that is hydrolyzed to ADP and an inorganic phosphate after incorporation into a growing F-actin filament. Figure 1 shows a deep cleft that separates actin into 2 main domains of almost equal size, each being composed of 2 subdomains, numbered SD1 through SD4. All subdomains contain a central beta-pleated sheet surrounded by a varying number of alpha helices. The deep blue coloring represents the bound adenine nucleotide, or ATP, which is located at the bottom of the cleft. Both the N and C terminus are located in SD1, and the peptide chain crosses twice between the domains at the bottom of SD1 and SD3. The amino acids are indicated in red, which are modified by bacterial protein toxins. Arginine 177, which is shown as R177, is ADP ribosylated by toxins. These toxins induce the depolymerization of actin. Threonine 148, which is shown as T148, is ADP ribosylated by photohabdus luminescens toxin, which is also known as TCC-C3. This toxin causes polymerization of actin. Lastly, various toxins catalyze actin cross-linked between lysine 50, which is shown as K50, and glutamic acid 270, which is shown as E270. As illustrated, these domains come together to surround the bound nucleotide ADP. The ADP remains attached to the actin subunit, whereas the inorganic phosphate dissociates slowly from the filament, generating two filament ends, with actin subunits differing in their bound nucleotide, either ATP or ADP. Actin polymerization proceeds until equilibrium is established between G and F actin. The concentration of the remaining G actin is the critical concentration of actin polymerization. During polymerization, ATP-bound G actin associates to the end containing ATP actin subunits, the fast-growing end, which is also referred to as the barbed or plus end. The other end is referred to as the pointed or minus end. The names barbed and pointed refer to the appearance of an actin filament when myosin S1 fragments are bound to it. After reaching equilibrium, actin monomers associate to the barbed end and an identical number dissociates from the pointed end. Under these conditions, and in the presence of ATP, actin subunits constantly associate to the barbed end and travel through the entire filament until they dissociate from the pointed end. This behavior is called treadmilling or actin cycling. The critical concentrations for the barbed end and the pointed end are 0.1 micrometers and 0.8 micrometers, respectively. Under polymerizing conditions, the critical concentration of polymerization is 0.2 micrometers, i.e., closer to the barbed end. As I mentioned toward the beginning, we will look at the research paper titled The Effect of Toxins on Inorganic Phosphate Release During Actin Polymerization. During the polymerization of actin, hydrolysis of bound ATP occurs in two consecutive steps: chemical cleavage of high-energy nucleotide and slow release of the gamma phosphate. In this paper, the effect of phalloidine and jasplikinolide on the kinetics of the inorganic phosphate release was monitored during the formation of actin filaments. An enzyme-linked assay-based spectrophotometric technique was used to follow the liberation of inorganic phosphate. Phalloidine, which is a cyclic peptide from Amanita phalloids, can bind tightly to F-actin. It stabilizes the structure of F-actin by reducing the dissociation rate of the monomers from the actin filaments. It has an equilibrium constant of 36 nanomolars. Jasplikinolide is another cyclic peptide that is extracted from a marine sponge. It also binds F-actin with high binding affinity and stabilizes the structure of F-actin. It has an equilibrium constant of 15 nanomolars. These two toxins bind the F-actin competitively and have local, short-range, or long-range allosteric effects on F-actin. Let's look at Figure 2, which shows polymerization curves. At the beginning of the experiments, the release of the inorganic phosphate from the newly formed 20 micrometer of 5% pyrene labeled F-actin was monitored in the absence of the two toxins, as indicated by the filled circles, and in the presence of 20 micrometer phalloidine, as indicated by the open squares, or jasplikinolide, as indicated by the filled squares. The vertical dashed line represents 180 seconds on the x-axis, and the horizontal line corresponds to 90% of the protomer concentration over total actin concentration. At the beginning of the polymerization, the inorganic phosphate release is not affected by the rate of association of the actin monomers. Polymerization tests on the pyrene-labeled actin reveal that, under the applied conditions, at least 90% of the actin monomers were incorporated into the actin filaments within 180 seconds, after polymerization was started. Moreover, the test completed in the presence of saturating phalloidine and jasplikinolide concentrations shows a small increase in the polymerization rate of the actin in the presence of any toxins, which is likely due to their nucleating activity. The ENS-check phosphate assay kit is fast enough to follow in real time the kinetics of the inorganic phosphate release. It also gives information about the total product released from the enzymatically cleaved high-energy adenosine-derived nucleotide. Figure 3 shows the change of the spectrophotometric signal with time for phalloidine and jasplikinolide. The amounts of the toxins were 0% for the filled circles, 25% filled squares, 50% filled triangles, 75% filled diamonds, and 100% filled stars. The increase of the absorbance at 360 nanometers was followed at 22 degrees Celsius for at least 1500 seconds in the presence of 20 micrometers magnesium-bound actin, as shown in Figure 3. The absorption transients were fitted with the combination of a single exponential function and a linear function. A single exponential function was used because the inorganic phosphate release from the F-actin followed first-order kinetics. The linear function was involved because of the slow steady-state ATP hydrolyzing activity of the filaments. The data shows that the inorganic phosphate release was 3.5 plus or minus 0.4 times 10 to the negative third Hertz for the actin in the absence of the toxins. Additionally, the total amount of inorganic phosphate released was determined by using a calibration curve acquired with a monopotassium phosphate solution as a source for inorganic phosphate. The amount of phosphate released during the fast phosphate release phase was 20.4 plus or minus 1.5 micrometers. This indicates that all the actin monomers are incorporated into the filaments and all the phosphates were released and detected by the spectrophotometric method. As stated earlier, the inorganic phosphate release measurements were repeated in the presence of phalloidine or jasplikinolide at 0, 25, 50, 75, and 100%, with the actin concentration kept at 20 micrometers. Due to the strong affinity of binding of these toxins to actin, with an equilibrium constant of 36 nanomolar for phalloidine and 15 nanomolar for jasplikinolide, more than 99% of the added toxin bound to the F-actin even at the smallest toxin concentration, like 5 micrometers. The polymerization test completed in the presence of saturating phalloidine and jasplikinolide concentrations showed a small increase in the polymerization rate of actin in the presence of any of the toxins, which again is likely because of their nucleating activity, as mentioned in Figure 2. These results demonstrate that both the phalloidine and jasplikinolide effectively blocked the inorganic phosphate release from the toxin-bound parts of the F-actin. It is important to note that both toxins affect the inorganic phosphate release similarly, which supports the assumption that, because of the high extent of the overlap of the binding sites of the applied toxins, their effect on inorganic phosphate release is very similar. In conclusion, the cytoskeletal protein actin is a frequently targeted substrate protein, modified in a manner that compromises its proper functions. Actin is constantly cycling between monomeric and polymeric states in order to fulfill its dynamic functions, including its diverse roles in innate and adapted immune responses. Therefore, disturbing that dynamic behavior of actin as achieved by ADP ribosylation will profoundly disturb the cellular level response to pathogen invasion. Notably, the bacterial ADP ribosyltransferases have been specifically tailored to modify residues like arginine 177, which are essential for its proper function, i.e. the ability to polymerize to F-actin filaments. One has to keep in mind that in most cases, the targeting of the cytoskeleton by bacterial protein toxins in effectors is much more complex. In fact, studies from recent years have shown that numerous pathogens produce toxins and bacterial effectors during host-pathogen interactions in a precise time and space-dependent manner to specifically support defined phases of the infection process. This explains the frequent findings that the same species of bacteria may produce different toxins and effectors, which cause polymerization as well as depolymerization of the actin cytoskeleton. Thus, bacteria are capable of modifying the cytoskeleton, thereby using the multitude of functions of the cytoskeleton for their own advantage, which, in my opinion, is quite fascinating, but also insanely scary. Thank you.